Hey everyone! Today we're going to explore something that you see in almost every living organism. DNA. DNA, formerly known as deoxyribonucleic acid, are the strands of molecules that travel through your cells containing all the genetic organisms making up the chromosomes in your body. DNA is formed in a double helix shape made up of 3 parts: a nitrogenous base, pentose sugars, and phosphate groups. The double helix is composed of nucleotides bonded to a sugar-phosphate background. You'll find 4 different types of base pairs or nucleotides in DNA. These 4 are adenine, thymine, guanine, and cytosine. The bases pair up with each other, adenine with thymine and guanine with cytosine. This is called complementary base pairing. What is complementary base pairing exactly? Well, as you can see in the pictures, adenine can form 2 hydrogen bonds with thymine. And cytosine can form 3 hydrogen bonds with guanine. That's why they can only be paired together. For example, if you tried to pair adenine and guanine, the compound would become too large and twist the nice double helix shaped DNA forms, and the hydrogen bonds that link these 4 different pairs would become too weak to be able to hold together a molecule that big. Now, we're going to take a look at RNA, which is short for ribonucleic acid. The difference between RNA and DNA is that the sugar in RNA is ribose, hence the R in RNA, whereas the sugar in DNA is deoxyribose, which creates the D in DNA. As you can see, their structures are very similar. However, deoxyribose has hydrogen where ribose has a hydroxyl. RNA is created through a process called denaturation. DNA is denatured when the strands separate. The hydrogen bonds that were holding together the nucleotide pairs break, while the bond that is holding the individual nucleotides to the sugar-phosphate background still stays intact. The process of breaking the hydrogen bonds between the nucleotide base pair in a double-stranded DNA requires energy. Because the hydrogen bonds are generally weaker than all the other bonds in the molecule, they will be the ones to break. And thymine is now replaced with uracil because it is complementary to adenine. Thymine is used instead of uracil in DNA because uracil is only efficient in RNA and not DNA. So, what exactly does RNA do? Well, protein production. DNA in the nucleus is copied by RNA polymerase, which reads the code and makes a complementary code called mRNA. RNA travels from the nucleus to the cytoplasm, where amino acids are located. The RNA code is read by a ribosome, 3 nucleotides at a time, linking together appropriate amino acids in a chain. The amino acid chain folds up into a 3-dimensional structure able to perform its function in the cell. You're filled with RNA and DNA, and these create genes which make you who you are today. If you have blue eyes, brown hair, if you're short or tall, all of that lies in the code that DNA and RNA create. You are who you are because of DNA and those little base pairs. So that's your quick rundown of DNA and RNA. Thanks for watching.